Good morning. Today I will discuss about my project, that is role of glycogenes in pancreatic cancer. So let us start with the background. So in the normal cells we can see this type of glycan profiles that has the complex N glycan, hybrid N glycans, and this normal O glycan structures. However, in the cancer cells these profiles get changes, and we can see some of the truncated O glycans and the more silated glycans that has this SLE antigen and SLEX antigen, and also this hybrid branched N glycan that has more branched than the normal complex N glycans. So this is the main reason we need to find out why this kind of changes occurs in the cancer cells. And one of the O glycans that is formed is shown here. That starts with this TN antigen, which is attached to the S and T amino acids, and when it gets react with this C1-GLT1, here it will form these kind of structures. However, in the case of cancer cells, the C1-GLT1 gets dysregulated, so, and because of the dysregulations, if the C1-GLT1s get down-regulated, you will see these silyl TN antigens occurred in the cancer cells. Whereas when C1-GLT1 is upregulated, we can see these kind of antigens, these silyl T antigens are more expressed in the cancer cells that are shown here in the form of truncated O glycans. And there are two studies that are published for the C1-GLT1 dysregulation of C1-GLT1 that can cause proliferation and metastasis of PDAC. First one is about this knockdown of C1-GLT1 that will leads to the metastasis, increased metastasis and progressions within the mouse compared to the normal pancreatic cancer cells that does not have C1-GLT1 knockdown. And these studies are done on KPC mice model. And there is one more study is done where it shows that this overexpression of C1-GLT1 will cause tumor growth and metastasis. And these studies are done in this NOD or SCID mouse. And this kind of mice has immune system compromised, or we can say the immune compromised mice models are used for these studies. So from these two studies we get an idea that this C1-GLT1 knockdown behaves differently in the presence and absence of immune system in mice, or we can say that it's important to have this proper regulation of these glycogenes. Otherwise, dysregulations of glycogenes will cause uncontrolled growth rate, and that might lead to the development of the cancers. So this is one of the motivations, that if this C1-GLT1 can cause dysregulations and leads to the more and leads to this more aggressive cancer behavior, then it is possible that other glycogenes might also contribute towards this proliferation and metastasis of pancreatic cancers. So in order to identify the role of those genes, we have created two libraries. First one is essential glycogenes libraries that contains around 42 essential genes that is necessary for the normal functions of the cells. Apart from that, we also have these O glycogenes, around 16 O glycogenes uh, that are also necessary to maintain normal functions of the cells. So dysregulations of this, so we will use this to knockout genes library, that is essential knockout genes library and O knockout genes library, to understand whether these glycogenes also has any role towards the metastasis or progressions of the pancreatic adenocarcinoma. So first we need to prepare a knockout libraries. So this is the plan that I used for this knockout library preparations. That is, first we take this pancreatic cancer cells. We use two different kind of pancreatic cancer cells. That is FG colo 357 and CD18 cell, CDC18 cell lines. And first we transduce this Cas9 with D tomato reporter. So once these cells are transduced with Cas9, cells will have the red fluorescence and they are red in color. And after that we will transduce these knockout gene libraries that has the CFP reporter. So now cells will now have, some of the cells will now also have this knockout genes transduced. So some of the cells will also show CFP positives. And thereafter we will use this Cas9G7 that has the CFP reporter. So what it will does is it will deactivate the Cas9 genes. So cells will not have now have this red color. Cells will be devoid of this red color and will only have this CFP color after the sorting. Now we are left with the libraries that is CFP positive and has the knockout genes, and all the genes that are used in the library are being knocked out. And this is CD18 library generation results that are prepared based on this protocol. First we transduce this Cas9, thereafter we transduce the CD18 libraries, and then we knock down Cas9 using Cas9G7. Now after bulk sorting we will have these CD18 libraries. And apart from CD18 we have the FG colo also. In this case we are not able to knock down Cas9, so we will have the libraries that has both the Cas9 and also the knockout genes. And after that, after we developed this library, we did the analysis to check the representations of the glycogenes, and we are able to see that in all the 4 cases the representations looks uh, evenly distributed. The representations of the guide RNA looks evenly distributed. In terms of the quantifications, we can see that in the O-glycans we have around 105 guide RNAs, and all the 105 guide RNAs are present in both the knockout libraries. And for essential genes and for essential plus scramble libraries, we have around 250 guide RNAs, and out of these 246 are present in CD18 and 248 are present in FG colo. And these are the guide RNAs that are not present in CD18 libraries, and these are the guide RNAs that are not present in FG colo 357 essential libraries. Once we have the knockout library prepared, now we need to do studies. So we started to check whether these knockout libraries will cause accelerated migrations or not. So this is the plan to check the migration assays and proliferation assays. We will place the cells inside this annulus ring, this PDMS fabricated annulus ring, and then we will allow the cells to grow for certain time, and after some time intervals we will fix the cells, and the area increase in the area we will consider it as migrations, and number of cells that has increased will be used to calculate proliferations. So, and apart from these migrations and proliferation, we will also check the impact of substrate on the hydrogel. And we used 3 different substrates: matrigel, collagen 1 and fibronectin. And this is one of the example where we do not see any migrations. That is when we used FG colo 357 cells and the material concentration is 100 microgram per ml. So in this case Right now it's not able to run this video. But I will tell you what is the final outcome. Final outcome and the initial outcome will remains almost same for these conditions, that is Fg colo 357 cells and the material is 100 microgram per ml. And this experiment is done in this case for about 6 days. And in the other case where here is the complete results where we did the proliferation assay on Fg colo wild type cells at different concentration of material, and we are able to see that at around 10 microgram per ml we see the maximum proliferations, and when it comes to the migrations we see migrations is also highest at 10 microgram per ml for the material. And when we used for collagen, we can see that for collagen the proliferation is highest for the 0 microgram per ml without any collagens. The proliferations is higher, but in the case of migrations, migration is higher at 10 microgram per ml. And this is the example for this fibronectin. Those results we will see in next slide. It's not running, but in this case the proliferations is increased to a greater extent, and the result we will see in next slide. I don't know why it is not running. This video is not running. So in the case of fibronectin, what we have seen is that at around 50 microgram per ml, the Fg colo wild type cells has maximum proliferations and maximum migration. So what are the final conclusions from this migration study is that for the wild type we can see at, for material we can see 10 microgram per ml we will get the more migrations, and for collagen we will get more migrations at around 10 microgram per ml, and for fibronectin we get around 50 microgram per ml. And after that we did the study on knockout libraries. Once we have this starting point at which concentrations we need to start for our knockout libraries, and in the knockout libraries we can see that for 50 microgram per ml fibronectin we can see the proliferations of the essential genes libraries is highest, and the migrations is also highest for 50 microgram per ml fibronectin. And for the CD18 essential gene knockout libraries, what we are able to see is that at around 10 microgram per ml, the 10 microgram per ml, we can see the proliferation is highest, and for the migrations also we can see that 10 microgram per ml, we can see the maximum migrations. So now we have the effect of stiffness. Now we know at which substrate concentrations the cells are migrating to a maximum extent. However, in actual conditions, cells migrations also depend on the stiffness of the substrate. For example, this is for examples we can see that when this integrins gets interact with the surface, it will lead to the change in the structure of actin, and this cell-cell interactions will also get affected. and all these interactions will trigger intercellular changes such as this actin polymerizations will occur. And here is the general timeline that is shown here. What are the changes one can accept in this time period? For example, cell signaling can be changes in minutes to hours. The gene expressions might take around days, and the cell behavior and the tissue formations will take around weeks. These changes will take around weeks. So we also need to check the effect of this different stiffness on these cellular behaviors, as then we will able to get what are the actual changes that is occurring in the patient. So cell migrations will also depends on the stiffness around the cells, and it is well known fact that tumor cells will change the stiffness by developing tumor microenvironment, and that will in return helps the cells in immune evasion and helps in maintaining the proliferations and migration properties. So right now what we have is that for these FG-Colo 357 cell lines, we know the substrate concentrations that works well for the migration is 50 microgram per ml, and for CD18 the 10 microgram per ml works well. We need to find the correct stiffness, and there are papers that has reported that in the pancreas the normal physical stiffness is around 1 to 2.9 kilopascal, whereas for the pathological conditions the stiffness increases and it is around 3.7 kilopascal. So this is what I am currently doing, is that to check the migration assay of FG-Colo essential gene libraries at this stiffness. This 2 kilopascal, 5.2, 11.6, 18.9 and 31.4. So this FG-Colo essential gene knockout on fibronectin at 50 microgram per ml concentrations. So this is how the cells looks like on the day one when we go seed the cells on the hydrogel that is coated with fibronectin. And in this case also we are not able to play the video, but the result shows that for the 5 days there is not much change occurs in this soft hydrogel, and this has a stiffness of around 11.6 kilopascal, and this has the stiffness of around 31.4 kilopascals. And here is the 20x image of how the cells are attached to the hydrogel that is coated with fibronectin. Here also we do not see too much migrations, and one of the reasons why we are not able to see too much migration is that the cells will not only depend upon the stiffness, but it will also depends on the surrounding environment. That is, there is one study that is published in the paper that when the surrounding environment is covered with this endothelial cells, in this case they have used UVEC cells, then they will see the more migrations. And so what they did is that they will see the more migrations. That the results are shown in this right hand side, where we can see that when the UVEC cells are present the migrations are more, when the UVEC cells are not present the migrations is less. So, and these studies are carried out for like 15 to 20 days. So what is the plan for my next studies? After I will check this results for like 1 or 2 weeks. If there is not much increase in the migrations, then I will plan to do these studies. That is, first I will take the annulus ring and place the pancreatic cells in the inside the annulus ring, and thereafter I will place UX cells in the outer side of the annulus ring. And then after 1 day I will remove the annulus ring and allow the pancreatic cancer cells to touch the endothelial cells, and thereafter I will start the experiment. I will start observing the migrations of the cells. So here is what is shown here, that at time t is equal to t1 the pancreatic cancer cells are low, that is has the smaller diameter, but at t is equal to t2 pancreatic cancer cells has high diameter. So this might increase the migration properties of the cells. So these are some of the further plans. That is, I will do migration assays with the endothelial cells around the hydrogel of stiffness 3.7 kilopascals. So I will do like one or two conditions below this and one or two conditions above this. And once we find out the optimum conditions for the proliferations and migrations, then we will use those conditions to do this spatial transcriptomics, where we will find out which is the glycogenes that cause maximum migrations and their cellular changes in terms of the pathways that are dysregulated. Once we have this in vitro results, then we will prepare individual knockout of that cancer cell lines and do the in vivo studies on the KPC mice model to validate the in vitro finding. And by comparing this in vivo results and the in vitro results in terms of the cellular changes and the migrations that we have observed, and we will be able to find out the glycogenes that has a more say towards this change in the pancreatic cancer cells, or that has a more say towards these proliferations or migrations in the pancreas, or that cause more proliferations and migrations. I hope you understood my project plans and my progress. If you need to understand more about this, or if you have any questions, you can write a comment. I will be happy to answer your doubts. Thank you for your time.